Assalamu alaikum class. We are going to start another topic related to our previous one. This topic is actually about some organelles of this bacterial cell. We have discussed something about cell wall of bacteria, but now we are going to discuss some more detail about gram-negative bacteria, and we will see some important things about gram staining as well. So. Before starting this lecture, please study the cell organelles of bacteria, uh, because without proper study or because proper understanding about the structure of the cell wall, you are unable to memorize the important components and important functions of cell wall. Okay, so let's start our today's lecture. We will focus on lipopolysaccharide, or you can say we will focus on the wall of gram-negative bacteria, especially in our today's lecture. And to understand the gram-negative bacteria or cell wall of gram-negative bacteria, first of all, we have to understand the gram-negativity and the gram-positivity. What is gram-negative and what is gram-positive? Why these terms are too much important? On the other hand, to study all these things, we will take introduction or small introduction of gram staining, a gram stain reaction. And before these things, we will review the structure of cell wall, which we have already studied in our previous lecture. And at the end of this lecture, we are going to discuss another structure of bacterial cell, which is actually cytoplasmic membrane. We will discuss some important function of cytoplasmic membrane also. So let's discuss some more some things about the cell wall. So we will take a short review about the cell wall of gram-positive bacteria. We have already studied that the gram-positive bacteria are composed with thick layer of peptidoglycan. Okay, so in in gram-positive bacteria, thick layer of peptidoglycan shows or helps in the identification. How is peptidoglycan helps in the identification in gram staining reaction? Special type of strain we known is that crystal violet stain. Crystal violet dye or primary stain binds strongly with the thick layer of peptidoglycan. Okay, if layer is thick, then it will bind strongly. Okay, and the it will not be removed after decolorizing with the alcohol. Okay, so it will remain attached with the till end of the reaction. So then it will appear blue in color. So gram positive peptidoglycan plays a very important role in the identification of bacteria, because crystal violet or primary dye strongly binds in with peptidoglycan due to its increased concentration. Okay, they usually appear blue in color or dark purple in color, because crystal violet shows blue color or purple color. So it binds strongly with the crystal violet. Then another important thing was the structure of peptidoglycan. Because in there are two components, peptides and glycan chains. Okay, so peptides are actually proteins or peptides. On the other hand, glycan chains are of two types, N-acetyl glucosamine and N-acetyl mimeric acid. You have already studied in your previous lecture about these two things, and please draw its structure again and again, because structure is very important. Of In our last stand-up exam, one mark question was just to draw the structure of peptidoglycan. ठीक है, so it is very clear in Levinson. So, when will you study these things? Please especially focus on the structure of peptidoglycan, because peptidoglycan is present in both bacteria, gram-negative bacteria and gram-positive bacteria. Okay, and we have already told you that as human cell wall is in human, no cell wall is present, and but in bacteria cell wall is present, and peptidoglycan is also present. So antibiotics, we can direct antibiotics to hit the peptidoglycan. So when they are programmed to hit only the peptidoglycan, then our body cells are safe, because our body cells does not do not have any peptidoglycan in any outer membrane. Okay, so our cells are safe, so that's why we can say it's a good target for antibiotics. Then another important thing about the gram stain. What is gram stain, and why gram staining is important? This staining procedure actually is developed in 1884 by the Danish physician Christian Graham. Okay, is a most important procedure in microbiology, because most commonly, most important and most economical method to classify bacteria into basic categories. Okay, so it separates most bacteria into two groups: gram positive bacteria as well as in gram negative category. Okay, so, gram-positive bacteria usually appears blue in color, and gram-negative bacteria usually stain red. The gram stain involves the following four steps. Okay, so in Levinson, there are four steps are mentioned. First of all, you use crystal violet to stain all cells. Then iodine solution will be added. Iodine to this iodine form complex with the crystal violet. So when iodine forms complex with the crystal violet already present or already bound crystal violet, then it forms CVI complex, crystal violet iodine complex, and this crystal violet iodine complex is removed by the decolorizer. If this complex is completely removed by the decolorizer, then it means the crystal violet was not strongly bind with the peptidoglycan due to the thin layer of peptidoglycan. Okay, so after addition of last reagent safranin or any other dye, then it will appear in pink or red color, because last or secondary dye will bind with the thin will bind with the cell outer membrane of the cell, because crystal violet has been removed already due to presence of thin layer of peptidoglycan. So this was the interpretation of gram-negative bacteria. In gram-negative bacteria, thin layer of peptidoglycan is present. As thin layer of peptidoglycan is present, so crystal violet is unable to strongly bind with the uh, peptidoglycan. So then, due to strong bonding with the iodine, a second reagent, due to strong with bonding with the iodine, it will be removed. This complex will be removed by the decolorizer or acid alcohol. Okay, so when crystal violet is removed, then blue color will be removed, and cell will appear red in color after saffron in addition. So, and these bacteria are actually gram negative, because they have thin layer of peptidoglycan. They are unable to strongly bind with the crystal violet. Okay, so these are actually basic things. In short, gram positive appears blue in color because of crystal violet, and crystal violet binds strongly because of the presence of thick peptidoglycan layer. On the other hand, in gram negative bacteria, no thick layer of peptidoglycan is present, so they binds weakly with the they binds weakly with the crystal violet, and it will remove later. It removes remove later by the addition of acid alcohol. So it will appear red or you can say pink in color. So this is actually a gram staining introduction. So we will go for practical of gram staining as well. Okay, so don't worry about this thing कि अगर आपको अभी कोई चीज़ में डिफ़िकल्टी है इसमें अभी, तो हम जब प्रैक्टिकल में इस बात को डिस्कस करेंगे, देन you will be able to understand it completely. So we are moving forward. So when gram negative will appear red or pink in color on microscopy, then why they then we should discuss some more thing about the gram negative. Gram negative bacteria don't have thick layer of peptidoglycan, a very thin layer of peptidoglycan is present in the cell wall of bacteria. Most of the area in the gram-negative wall is covered by the outer membrane, which is actually lipopolysaccharide membrane. Okay, so most of a thick layer of lipopolysaccharide is present in gram-negative bacteria outside of the peptidoglycan layer. So these lipopolysaccharides are usually considered as endotoxins because these are actually because these lipid molecules act as toxins for our body, because these toxins can cause fever, these toxins can cause shock condition in body, and if shock condition remain for several days, then a patient can lead towards the death as well. So you have already studied about the septic shock, and septic shock is associated with the toxins, okay, or toxins or sometimes bacteria as well. So in a nutshell, endotoxins are lipopolysaccharides are endotoxins. So endotoxins are integral part of cell wall, and they are involved in the fever. They are involved. They are major cause of fever and shock in the human, or you can say a septic shock as well. Lipopolysaccharides have the three basic units, three subunits. You can say phospholipid molecules. These are phospholipid molecules are also known as lipid A molecules, and these lipid A molecules are actually main part of the endotoxins of the gram-negative bacteria. Polysaccharides attached with the another part is 5 carbon sugar, which are actually binds with it. These are actually core polysaccharides. Second part are a core polysaccharide of 5 sugar, okay, linked through ketodeoxycutulonate. Okay, another compound ketodeoxy KDO, ketodeoxycutulonate to lipid A molecule. Okay, so phospholipid 5 carbon 5 sugars for 5 polysaccharide sugars bind with the lipid A molecule with the KDO, and then another part is an outer polysaccharide. Okay, and another outer polysaccharide consisting of up to 25 repeating units of 3 to 5 sugars. Okay, मीन्स इन लास्ट जो ये अब तीसरा स्टेप है पोलीसेक्राइड्स विद 5 शुगर, इसके रिपीटिंग यूनिट्स हैं यूजली यूनिट्स आर 3 to 5 यूजली 25 इन नंबर्स, ठीक है. द आउटर पॉलीमर is the important somatic or O antigen in outer. These polysaccharide last जो third polysaccharides हैं are actually outermost part of the polysaccharide structure. So these polysaccharide structure are also act as an antigen because these antigen are also known as somatic antigen. Okay, it's the nature of gram negative bacterial cell wall. So if we are able to identify these antigens, O antigen actually, then we are able to understand the strain of bacteria, its disease mechanism, and we can also it will be also helpful in the selection of antibiotic to kill itself. So antigen, O antigen is much important, and uh, apart from help in identification, these O antigens also an important cause in are important participant actually in disease cause, in disease causing in host. So in humans, O antigen can also cause disease in humans. So O antigens are basically are one of the virulent factor of the gram-negative bacteria. Okay, so important in identification of bacteria. I have already told you that these are it is important in the identification of bacteria. So another important topic is about the cytoplasmic membrane. Usually, it remain sometime unimportant, or sometimes we are not focused on this topic. Cytoplasmic membrane, what are cytoplasmic membrane? Just inside these are membranes which are present exactly just inside the peptidoglycan layer. Okay, so after cytoplasm and inside of the peptidoglycan layer, another layer is present which is known as cytoplasmic membrane. If we talk about the composition of the cytoplasmic membrane, cytoplasmic membrane is composed of the lipids bilayer model. You have already studied lipid bilayer model. So phospholipids bilayer of phospholipids is present in the cytoplasmic membrane. So basic composition of cytoplasmic membrane is lipid bilayer, and location. You should very well know about the location of this component. So this organelle or component is present inside, just inside the peptidoglycan layer. Okay, chemical similar to the eukaryotes. Chemically, cytoplasmic membranes are actually similar with the eukaryotic uh, membrane, because in eukaryotic membrane phospholipids are present. Okay, no issue. Lipid bilayer model is also applicable in the eukaryotic cell, but in a eukaryotic cell there is another or exception is the presence of sterols. Sterols are not actually present in the cytoplasmic membrane of bacterial or prokaryotic cell. Sterols are present just in the cytoplasmic, eukaryotic cell. So here is an exception. All other composition is actually same. Importance of cytoplasmic membrane means we are going to discuss the function of the cytoplasmic membrane. Cytoplasmic membrane is actually involved in the active transportation. So it has been the active transport of molecules into the cells. Another function of cytoplasmic membrane is the energy generation mechanism. These membrane have special molecules or special type of special composition, because oxidative phosphorylation mechanism of oxidative phosphorylation runs on it. When mechanism of oxidative phosphorylation runs on it, then it produces it they produce after oxidative phosphorylation, they produce energy for the cells. So you can say cytoplasmic membrane is involved in the formation of energy due to oxidative phosphorylation on it. Okay, so as this membrane is present just inside of the peptidoglycan or just inside of the cell wall, then it also provide precursors or some basics unit for the manufacturing and repair of this cell wall. Okay, so then this cytoplasmic membrane have special type of molecules which are involved in the production of many enzymes. Okay, so these enzymes perform some major metabolic functions for bacteria, and these enzymes are also helpful in the defense of bacteria. These enzymes are also involved in the resistance against drugs. Okay, so and these are also a site for the toxin production as well. So there that was some important function of the cytoplasmic membrane. ठीक है, तो आप लोगों ने इसको भी इग्नोर नहीं करना है, इसको भी आपने उसी तरह याद करना जिस तरह बाकी ऑर्गेनेल से. थैंक यू. सो if you have any question you can discuss me on WhatsApp as well. Thank you so much.